He's currently director of the small grains breeding program and assistant professor at the University of California, Davis. Xiaofei has over 15 years work experience leading breeding across 4 major crops: intermediate wheatgrass or Cassava here at University of Minnesota, sweet potato, cassava, and bread wheat. This morning, I also learned he's responsible for barley, oats, triticale, what else? Did I miss anything else?
I think that's it.
Before UC Davis, he directed a global cassava breeding program at the CIAT, International Center of Tropical Agriculture in Colombia. His current research at the UC Davis focuses on increasing yield beyond the Green Revolution and to develop varieties with superior nutrition and disease resistance. We're excited to hear his talk of crop improvement for evolving challenges. Thanks, Xiaofei.
Thank you much, Julian. So yeah, actually, I'm very excited to return to University of Minnesota. Actually, my career in plant breeding started from here. So that's about 13 years ago in 2012. I joined Jim's lab, work on intermediate wheatgrass. I learned from breeding from Jim and also Rex Bernardo here. So very excited to come here to share some stories based on my experience on wheat and also the cassava. Try to see how we breeding program and breeder try to deal with the challenges from farmers, from consumers, and also from the environment. So I think today, so if you don't remember anything else, I hope you remember this triangle. So any breeding program, we deal with the challenge, the demand from farmers, from growers, and also we need to try to minimize the impact of agriculture on the environment. So, and for breeding program or breeder, we have the toolbox. This is what I learned from Rex Bernardo. So that's the plant breeding triangle. So to deal with those challenges, we need to either discover new germplasm, or we can create a new germplasm, new variation. And also we need to develop or implement the tools. Then we can improve the germplasm, or we can integrate those germplasm into our breeding to develop new varieties. And also nowadays is not only breeding programs, so we need to have multidisciplinary team work together and try to fix the challenge, or to address the challenge, or fix the problem together. So before jumping into cassava and wheat, I would like to talk about, can introduce myself a little bit and also the program. So I joined Davis last year in March. So my predecessor, Jorge Dubkowski, so he did not retire, but he has his own funding resources, HHMI. So he is more independent, and he now shift his research into basic science. Is mainly on spike development. It's mainly about flowering, about how the spike structure formed, and I'm mainly take over the breeding responsibilities, work on red wheat, durum wheat, barley, oat, and triticale. So we have two excellent field breeder. So one is Josh, is on the right, on the right side. So he is leading, actually in charge of triticale breeding, but also work with me on wheat breeding. And also we have another field breeder, Alicia Del Blanco. So she mainly leading the barley and oat variety development. So this is kind of the team. Before joining UC Davis, I work at CIAT, it's an international center. If you not heard about CIAT, I believe all of us know CIMMYT. So where Dr. Noam Bullock implement Semidorf gene to initiate the Green Revolution. So CIAT is a sister research center in the CGIAR system as CIMMYT. So at Seattle, so we have crop research. So there are 4 major crops: cassava, rice, beans, and tropical forage. And also have other research area like climate change, like biodiversity, and also even food system. So based on my knowledge or my experience working at Seattle, so now I'm organizing a study abroad course, try to help students to develop the global perspective in agriculture. So that's what happened next summer in August. If you have any student interested in this area, please send them to my way. So we also can so this program not only for UC students, is kind of for all the students in the US. We can find a pathway to work that out. So this is a picture of the Cassava program at Seattle. So that's about 2 years ago during the annual retreat. So we have a pretty broad research area. So that's we have the multidisciplinary team. So we have scientists working on the plant pathology, even food science and seed system agronomy. So there are about 1/3 of the people work on breeding. So that's kind of my area. So we are an international research center. So our mission is trying to serve small field farmers in developing countries, especially in tropic countries. So our funding is about 20 to 30% of the budget is from the core funds from the CGIAR, and all the others are mainly from bilateral project. So it's mainly like probably some of you know the Next Gen Cassava project, mainly funded by Gates Foundation, and also we have project funded by USAID and some project funded by Australia government. We also have a collaboration with the industry. I don't know whether I have probably not. The ingredient is a company support us to develop the vaccine cassava. So and also you can see how it plus there also support on the by fortified cassava there. So cassava. So come back to cassava. Cassava was domesticated in Latin America. So in the Amazon region. So at Seattle we have that location kind of benefit. So we have the largest cassava collection in the world. So we conserve about 6,000 cassava accessions there. Nowadays, cassava has been grown mainly in Latin America, in Africa, and in Southeast Asia, mainly in the tropical countries. And Nigeria in West Africa, so that's the largest cassava production country. So cassava in Africa is mainly for food, but in Southeast Asia is mainly for starch. So cassava is very well known because of its drought tolerance. So it takes about 10 to 12 months for cassava to develop a very large storage root. So it survives through the rainy season and dry season. Especially during the dry season, cassava can drop all the leaf. And when the rain comes, they can redevelop the leaf and then continue to accumulate biomass. So this is kind of how cassava looks like in the field. It's kind of a bushy crop, but if you dig it, you can see the roots. So this is probably a very extreme example. The root size is that big. And in cassava roots, about 85% of the dry matter is starch. So that's why it's very important starch crop in some countries. And also using cassava flour, we can produce different food product. So on the top, that's gari and fufu. So that's mainly in West Africa. So people ferment cassava roots and then dry it, produce flour, so and then can be processed into this kind of fermentation related food product. It has some special sour flavor there, and for sure it also can produce starch and we also can dig the roots and boil it and eat it. It's pretty plain, don't have much flavor. I think this is why it can be used, can be considered as a staple crop like wheat and rice. There's not much extra flavor there. Probably some of you like bubble tea. If not, maybe your kids might be love bubble tea. So the bubble there is mainly from the cassava starch. And also, if you are interested in more recipe, so the Seattle food scientists they develop a small booklet is basically how to use cassava flour to develop a different product. You even can use it to make ice cream. Okay, so again, so cassava is clonal propagated crop. So after farmer harvested the roots, he can collect all the stems and stack them together. So once the rain is come, the soil moisture is good, he can cut the stem into about 20 to 25 centimeter long sticks and put it in the field. So the next production cycle will start. In terms of genetics, cassava is pretty simple. It's diploid. The genome size is about 800 megabases. So it's relatively small. It's about the size of one chromosome in weight. Okay, so that's also make us affordable to sequence the whole genome and to do some discovery there. And cassava is cross-pollinated, so it's highly heterozygous. So if we cross any cassava parents in the F1 stage, we're going to have broad segregation. So, but this also give us some trouble in breeding. And also cassava, since it's highly heterozygous, it have pretty high genetic load, have quite a lot. The deleterious mutations make inbreeding a little bit challenge. But cassava is self-compatible. We can generate seeds. Again, as I mentioned, so the growth season is pretty long, 10 to 12 months. So in general, we only can have one generation a year. So the breeding process is relatively slow. So come back to the challenge triangle. So we always know farmers want the variety without any disease, have high yield, good yield stability. And for consumers here, mainly about high beta carotene, that's pro-vitamin A. And for processors like ingredient, they really like waxy cassava, so which can provide some special starch functionality. So in their product, and for fresh consumption, so consumer like the good clean quality is the taste or the structure in the mouth is more smooth, the better. We don't touch much about the environment side in terms of cassava. So for all these traits, the consumer needs, the farmer needs, it's very hard to put all these traits in one variety. So at Seattle we have different breeding pipelines to focus on different market segments. So we have a high dry matter yield is mainly to meet the starch industry need, and we have fortified cassava is for high beta carotene, and we have the good clean quality cassava mainly for fresh market. With the waxy cassava is mainly for its a special starch functionality, and the processing cassava is mainly in West Africa, is mainly about the fermentation related product. Okay. I want to highlight all these traits we are working on now today in cassava, all are from the gene bank. So I think this is kind of the advantage of Seattle breeding program. We can access the germplasm, we can identify the new traits and bring that to breeding. So here I want to highlight several samples and how the Seattle breeding program discovered the new diversity and bring it to the breeding program. So first is by fortified cassava with high beta carotene. So this is about 15 or 20 years ago. It's supported by Harvest Plus. So my predecessor at Seattle, Dr. Hernandez Bajos, So he started to work on the light yellow cassava in the gene bank and then make crosses and try to improve the beta carotene content. So you can see in the past 15 years, we made significant progress in terms of beta carotene content in cassava. So, but recently we noticed that when we plant this improved cassava in beta carotene into the targeting environment, we observed the low germination and the low germination or the low sprouting ability. So you put the stem in the stick in the field, but not all of them sprout. And also the queen quality is not that good. So the consumer and farmer not adopt this new variety. So then we work with food scientists to develop a method. It's called water absorption based method to evaluate queen quality. So you can imagine the cassava roots you put the cassava roots in water, and during the boiling process, the root absorb the water. So those roots absorb water quicker, can be ready faster. So this is kind of the water absorption value. So this is about the water absorption value, and so here is about the queen time. So usually the large water absorption value, and it means it's be ready faster. So this is kind of the way we can measure queen quality. We can also kind of select for high beta carotene together. We improve both traits together. So another example about gene bag material is waxy cassava. So I think this is also initiated by my predecessor, Cassava. So I believe at the beginning, so we are not quite sure whether there is this kind of mutation in the gene bag. So he just started, and we know waxy starch is recessive trait. So cassava is heterozygous. So the waxy starch only show up in S1, in S1 progeny. So he screened about 20 to 30 S1, 20 to 30,000 S1 clones. He identified several clones showed this waxy starch. And even now, we don't know why this resistance is. We don't know its biological means for cassava have this kind of mutation, but it is there. So the blue one, so that's normal starch. The brown one is waxy. So it's very easy to screen. You just kind of stain it, and then based on the color, you can identify. So after we identified that starch, the food scientists recognize the cassava, right? Cassava waxy really have some pretty good feature advantage there. So first, the paste is very clear, and also it's very tolerant to the freeze and thaw cycle. So cassava starch is here, so it's much better than the other kind of starch. Is more tolerant in the frozen food product. So this is kind of the major reason ingredient is very interested in waxy cassava there, and then the later ingredients support us to continue develop waxy cassava for high yield and for high starch content. So, and also, so this is kind of always the story. We find some new traits, we introduce that in breeding, so there is always some problem. So again, so we find that there is the waxy cassava is also have the sprouting issue. It's not sprout well. So we are not quite sure it's because of the pleiotropical effect of the waxy starch or because of different gene bag. So we are still working on trying to figure it out. But for this problem, we work with seed system scientists. So we optimize how we manage the stem, how we manage the stick after harvesting. So in production, we can solve this problem. It's not based on genetics. It's kind of based on mainly how we manage the seed system. But for sure, in breeding, we are continuing to work on that, work on improve the germination. The next is about the disease. So again, so this is all So in cassava, there are this kind of disease in different continent, but we find the resistance from the gene bank. So here I want to highlight one is the cassava brown streak disease. So this disease is only happening in Africa, but in Latin America, germplasm or in the gene bank, we identify the resistance. So this is a collaboration with a plant pathologist in Germany. And after we identify the resistant accessions, then we using culture mapping or tissue mapping, we identify the major genome loci associated with the resistance. And then now we have the marker available. We can integrate that into breeding. So for whitefly, also quite a similar story. We find resistance in the gene bank. So I think the major question or the problem for cassava is highly heterozygous. We cannot do a backcrossing based trait integration. So we have to do a kind of recurrent selection, try to improve the population cycle by cycle. So even we know there are major genes controlled that traits. So this is kind of the typical breeding scheme in cassava. Yeah, we also use cross, and I mean we cross two parents to separate the population. So because the parents is heterozygous, in F1 we have broad segregation. And in the following years, we just use clonal propagation, and then we evaluated the same genotype year after year. And after several years, we are confident about the performance. Then we cycle back at parent. So from crossing nursery to crossing nursery is about 5 years cycle. So based on my learning in genome section at University of Minnesota in intermediate wheatgrass, so I still believe so. This is a good strategy to implement, and so that is we use a subset of the breeding population to train the germ section model, and then to predict the performance of the remaining in the population. So the close relationship between the training and the breeding population give us a kind of high predictor ability. So we use the similar strategy in cassava. So the only thing is we still rely on the field evaluation, try to trim down the population size. So we cannot afford genotyping 10,000 lines. So after 1 year field evaluation, we discard all the poor performance lines, and we genotype the breeding population and bring the genotype data to the model, and then we can calculate the GB weight, and then to select the parent. Again, the predictability is moderate and high. So this is mainly about the traits farmer cared, the yield, the plant architecture, and also the dry matter content. I think so. This is, I mean, using a subset of the breeding population predict the remaining still works pretty well. So this is kind of the start point of how we implement genome section in cassava. So the next, I want to quickly talk about the CEAT cassava breeding program. We initiated the hybrid cassava breeding. So I believe some of you might heard about hybrid breeding in potato. So they use diploid and then develop an inbred progenitor. So here for cassava, we also have quite a similar pathway. So not compare with potato, so just compare with corn. So cassava is quite similar as corn. So both are kind of starch crop, and both are diploid, and both are cross-pollinated, and both are self-compatible, and both show pretty good inbreeding depression. But for cassava, it's clonal propagated, so we don't need the hybrid seed production. So this is kind of also an advantage if we can in cassava, if we can develop hybrid breeding program system. But if we look at the yield compared with corn and cassava, not to mention the right side, so the corn in the US, if we only compare the corn and cassava in tropical country, so we can see corn yield increase significantly, but cassava yield not increase much. So the exception is in Thailand is mainly driven by the starch industry, but in the last 10 years, the yield is not increased much. So I think it's mainly because cassava lack the inbred progenitor, not able to systematically use hydrolysis, and not able to do backcrossing efficiently. So I think so mainly because of this, we are thinking, so cassava has, it's doable for cassava to develop inbred progenitor to transform the cassava breeding into a hybrid breeding system. So there are several technologies available can help us to do that. So first, we have the flower inducing system. So we use photoperiod extension and hormone. We can generate we can generate a flower, and then we can produce seeds. So you can see the blue dot. So that's the natural light, and the orange one is photoperiod extension. So for some clones, for some parents, we can significantly reduce the flowering time, and so that make cross doable. And also, there's a company in Germany, so they provided the service to develop protocol for double haploid. So before I left CEAT, we developed a project funded by Gates Foundation to work with this company to develop a protocol for double haploid cassava development. I think the project is still ongoing. So I think based on this improvement and this technology, so for cassava hybrid breeding, so we mainly focus on this research area. So first, we want to understand inbreeding depression in cassava, and also we are going to develop inbred progenitor, and also we have a model try to improve the population to minimize the inbreeding depression, and we are going to develop heterozygotic groups, and then we can systematically use hydrolysis. So we started from a small population. So this is 5 family from 3 progenitors. So we have three S1 family and 2 F1 families. So we have the genotype data, and also we evaluated the performance in the field. So this is mainly also collaborated with BGI, is a sequencing company. So we work together to assemble the genome of the parent, and also we predicted the deleterious mutations, and also estimated the genetic load. And based on this population, we identified the associated loci related with different traits. So then we try to link the genetic loci with deleterious mutations and inbreeding depression. We try to form this kind of story to understand inbreeding depression in cassava. We are still working on that, but here is just some results we have now. So first, we assembled the genome of the 3 progenitors. You can see so this is just one progenitor, the 2 haplotype. So the assembled quality is much higher even than the current reference genome. And so this is the 5 families, 3 S1 family on the corner, and 2 F1 family kind of in the middle. So we use 10 times whole genome sequencing to get the genotype data. So again, the genome size is only 800 megabase. It's doable to do the whole genome sequence to get more data there. And in terms of the field evaluation, we have pretty good repeatability or broad sense heritability in our evaluation for all these different traits. And then we kind of did a structure mapping or linkage mapping, try to identify some genome loci related with yield or related with dramatic content, plant architecture. So this is just some example about the genome loci related with yield or yield related traits. there. So there's just another one example is about the dry matter content. So this is a gene loci on chromosome 3. So it's associated with dry matter content. So at the end, we narrow down that loci into a candidate gene. So we find this is because of structure variation. So in the figure B, you can see on the top, that's one haplotype. There's only one copy of the candidate gene. But at the bottom, the orange one, so there are several copies of the candidate gene. So that's the orange box there. So after we analyze the phenotype data, we notice this orange box with several copies of the candidate gene it show low dry matter, but those one with only one copy show high dry matter. So this candidate gene in Arabidopsis, if this gene mutant, so it has high dry matter. So that's kind of aligned, high starch. So it's aligned well. So if we have less copy, it has high starch or high dry matter. And also we try to link this loci with deleterious mutations or genetic load. So here is kind of the correlation between the dry matter content with the homozygous burden. So there is negative correlation. So that means if there's more inbred, so the dry matter will be lower. So this is kind of the story we are trying to frame. So we identify this loci and try to understand why, and then to link is with genetic burden with the deleterious mutations there. So another work we are doing is try to initiate and develop the hybrid breeding. So we started with selfing about 30 elite progenitors. So we developed S1 families, and start from there, we develop a 5-year roadmap. So this is mainly in the next-gen Cassava project frame. So it's pretty busy work, but there are mainly four modules. So first, we are going to evaluate the S1 family, try to understand the inbreeding depression in more diverse Cassava parent. And then we also have model try to continue selfing and try to develop inbred progenitors. But we are not, because of the heavy inbreeding depression, we are not quite sure how far we can go. So we have another module. So we are going to cross those S1s, and we are going to do recurrent selection, try to minimize the effect of inbreeding depression, and then hopefully in the progeny we can go further. And we also have the fourth module is to do test cross and try to figure out the heterotic groups. So at the end, so this is our dream. So we are going to transfer Cassava breeding from selective breeding to this kind of hybrid breeding. So basically we are going to have one foundation population. We are going to use inbred progenitor. We are going to systematically use heterosis, and for all the other market segments, we are trying to use treaty regression. Then we can all this breeding effort will build on this foundation population there. So so this is the time when I was at CED. So this is the system we are developing. I'm not quite sure after 2 years whether this is still going or not, but that's kind of the design I have there. So kind of summary in Cassava. So how we deal with these challenges. First, we understand the needs, and then we kind of bring, basically, especially in Cassava, we bring this new diversity from the gene bank to the breeding program to figure out, to try to address the different problem in different pipeline, and we develop new tools like phenotyping tools, like implemented germ selection, and even we go further to redesign the breeding scheme using hybrid breeding system. So quickly jump into weight. So I just started last year, not much solid result to present, just share some research. We are some research we are doing, and hopefully we can find some shared interest and develop some collaboration or have more conversation on this. So come back to this triangle. So in weight, so first, let's look at so this is mainly about the wheat production in the US. So we can see the acreage in the US going down pretty quickly, and now it's a little bit stable. So we know wheat in quite a few states is not a major cash crop. It's mainly become a rotation crop. So farmer probably will not willing to put high input into the wheat production. So the resource use efficiency is important for wheat variety development. And in terms of drought, so this is a figure in California. If we look at the last 20 years, so we have more frequent and more severe drought. So this is also kind of reflect to the farmers' needs. So California Wheat Commission asked us to develop drought tolerance with variety. So this is some research area we are working on. And for disease resistance, so strep rust is the major disease in California. We don't have, so the other diseases not happened often. So, but strep rust is every year we have a pretty stable strep rust pressure. So this so there are two major genes, WR5 and WR15. So that have been protecting our wheat production for about 20 years. So this year, WR15 have been broken down in the UK in large scale. So this is really kind of raises the alarm for us. We need to continue search new resistant gene and to integrate that in our breeding program. So in terms of consumers' concern, so this is the curve of the wheat consumption in the US. So there are mainly two waves of concerns from consumers. So first is about in 2000. So consumer prefer the low carbohydrate diet, so require higher dietary fiber. And so another one is gluten-free food. So kind of reflecting on this demand on these challenges. So this is kind of the research area we are doing or we plan to do. So we are developing varieties with high amylose starch, and also we are doing some research on reduced gluten allergenicity, and we continue search a new strep rust resistance and try to develop tools to evaluate the drought tolerance and also nitrogen use efficiency. So here I will mainly kind of talk about two research or two projects we are doing. First is to replace the green revolution genes, and so the other one is a tool development. We are using genome-based phenotyping, try to evaluate nitrogen use efficiency. So for the green revolution genes, it has been used in breeding program for 50 or 60 years in about 80% of the released variety. But we know so this gene mainly works well in high input system. So this gene is a global depressor. So in high input system, it can control the plant high well, reduce lodging. But in medium input or in low input system, so the low signal vigor or the low biomass makes the plant very sensitive to environmental stress. And also this gene is affect the nitrogen use efficiency. So there are quite a lot of studies trying to identify the alternative Semmendorf genes. but so far there's no one have been widely deployed there. So and if there is, so here we mainly focus on the two genes so discovered or studied by Jorge Dubukowski. We continue work on these two genes. So first, the plus A1, the plus one. So this is from file mapping to clone the gene for IC25. So it's a transcriptional factor. So if this gene mutant, so it will show the Semmendorf phenotype. The AP2L2, so this is from reverse genetics. So when they study the microRNA 172 and the Q genes, they identify another gene is AP2L2. So this gene is affect plant height and also affect the spike architecture. So this gene is regulated by microRNA. So if we mutant the binding site of the microRNA, so the expression of this gene will go up and it will show the Semmendorf phenotype. Okay, so we have so there is already some result to show. So this is the CRISPR mutation in triticale under the green revolution background. So it still reduces the plant height, improve lodging, and there's no significant effect on yield. So this is the result about plus one. So again, it's under the green revolution Semmendorf gene background. So it's also significantly reduced plant height in different lines, but not a significant effect on yield. So now what we are going to do is we try to bring this gene into the non-green revolution background. So this is the population we are developing now in BC4 stage, basically for plus and for AP2L2, the similar story. So we created this combination of mutations and under non-green revolution background and green revolution background. We are going to have a field trial. So this year we are increased seeds, and next year we are going to have multi-environmental field trial. So this to evaluate the effect of these mutations in allele background, and to really to see the potential of these two genes and which allele can be used to replace the green revolution genes. And at the same time, we are building the population from 4 parents. So these let's see. So these two are the allele, the backcrossing lines in allele background with the mutation of the two genes, and also the other two are the allele parent. So we are doing this is mainly because green revolution gene have been used in breeding program for about 56 years. So breeding program have been manipulated the genetic background, try to feed the green revolution genes. So if we bring a new gene into the system at the beginning, probably it will not work. So we are developing this population. We try to optimize the genetic background through recurrent selection, and hopefully after 2 or 3 cycles, we can reach the level and the background can match the new Semmendorf genes. So this is also aligned with a germ section breeding scheme proposed by Dan Rancy, a professor at UC is mainly we are going to perform within family recurrent germ selection using this population, try to manipulate the background. I mean, before these slides mainly talk about the EMS mutations. So all these mutations can be freely used in variety development. But there's only one, so for each mutation, there's only one type. But CRISPR gives us an opportunity. We can create a different type of mutation in the microRNA binding site. So we expect this different type of mutation will have different effect on the plant height. So at the end, we hope we can create a series of variation or alleles can have different plant height. So maybe some allele can fit in California, some allele can fit in Minnesota. So this is so now we are we are developing the homozygous mutations and remove the Cas9, and then we can put that into the field to evaluate its effect. Also, we are going to bring that in the non-green revolution background to see their potential in replace the green revolution genes. So this is mainly about our work is on the alternative Semmendorf genes. So we try to at the end, hopefully we can find some allele or some genes which can still help reduce the plant height, reduce lodging, but also have higher seedling vigor and more tolerance to environmental stress, better nitrogen use efficiency. So the next we'll be talk about tools we are developing. We mainly work with small grain agronomist, Dr. Mark Lindy. So I believe a lot of we know in June we have if we attach a camera with on the drone, so the drone can capture the different spectra, and based on these different spectra data, we can evaluate the the stress level of the plant, or we can evaluate the growth stage or the growth status of the plant. So there are quite a lot of studies try to using drone data, the spectral data to predict the yield. But there are a lot of it's a lot of uncertainty there. It's mainly because if we think about the theory, the source, the flow, and also the sink, so the drone can only see the source. But if we want to, so there is a flow between the source and the sink, but this flow is heavily affected by different environment, by the rainfall, by the temperature, by the soil. So a lot of these prediction using drone to predict yield not work well across location, across years. So learning from that, so for us, we mainly kind of using drone data try to characterize the growth of the weight. So we collected the data about early stage vigor and also about the terminal stage drought tolerance and plant height and nitrogen. So here we'll be mainly kind of focused on nitrogen here. So again, collaborate with Mark Lindy. So he in charge of the variety trial and University of Minnesota. So every year he has the high nitrogen and the low nitrogen treatment. So he collected the drone data and also he collected on-ground biomass and the nitrogen concentration in the plant. So using that about 5 years data, he put together and developed this machine learning based prediction model. So he can predict the biomass and the nitrogen concentration well. So his main focus is try to evaluate the farmer's field, to fly the drone through farmer's field to tell farmer whether you need to apply more nitrogen or not. But for us, as a breeding program, we're mainly interested in whether this model can help us to distinguish the different lines. So some line might have more efficient in uptake the nitrogen. Some line might less efficient. So this is roughly the schedule we fly the drone through our field is every 2 to 4 weeks, and so this is the field at Davis is about 20 acre size. So we plant all our trials in one field, different crop. So just use one flight, we can cover all our trials in that location. Is about 1.5 hour flight. So we have accumulated about 2 years data, about 10,000 plots. So now we are still working on how to extract the data from this drone camera to see how this data can help us to make better breeding decision, evaluate the early stage vigor and terminal stage drought, and also nitrogen. I'm still analyzing the data. Here is just show one figure. Let's try to focus on the nitrogen use. It's mainly from the model developed by Mark Lindy. So we can see we have a moderate or high variability in terms of nitrogen percentage or biomass or nitrogen uptake. But we are still in the process in evaluate whether this prediction we claim is nitrogen concentration, we claim is nitrogen uptake, whether is real or not. So in terms of the heritability is good, we can capture some genetic effect, but whether it's true nitrogen uptake or not. So this is something we have a graduate student is collecting the ground data about the nitrogen concentration, about the biomass, and then we are going to validate whether this prediction is correct or not. So again, so this is ongoing, but I'm pretty optimistic. Either if we cannot use his model, but probably we can use our breeding data to update his model. So this is ongoing work. Briefly about strep rust, so we mainly evaluated the germplasm from USDA scientists. So Xiaofei Zhang, I believe quite a few of you maybe know him. So he is from pathologist at USDA. So we evaluated some landrace he identified for strep rust resistance, and also Stephen Xu provided the global Durham panel. So we evaluated the strep rust resistance, find pretty good amount resistant lines, and also he developed synthetic wheat. We also evaluated those population too. And for nutritional value, we made work with Jorge Dubkovsky to continue some previous research. And in terms of gluten allergenicity, kind of briefly, we mainly work on the three locus where glandian gene located. So the glandian gene is always located as cluster in that 3 chromosomes there. I mean, in this six chromosome. But in these 3 chromosomes, there is more immunodominance epitopes there. So then we focus on these three loci. We use the material created by USDA scientists. They use radiation mutation and created the deletion of that loci. So now we are developing near isogenic line to try to evaluate the effect of this gene on quality traits, on agronomic traits, but it's not that easy to evaluate the gluten allergenicity. But so this is so this is something we are working on. We are moving forward. In terms of the high amylose starch, so again, so this is some previous work by Jorge Dubkovsky. I believe in Minnesota we also have this material here. So there are 6 genes, ABD. On each there are 2 genes control the amylose production of the amylose pathway there. So we have 5 mutations. We have the mutation of these two genes, and 1 gene on the D genome. So with these 5 mutations, we can have increase the amylose content from 20 to 40 or 50%. But this since we changed the starch, the starch composition in the plant, so it has yield penalty about 50 or 20%. And also here I have figure about the quality is also affect the bread making quality. So now our breeding is mainly kind of conventional breeding using recurrent selection to manipulate the background and try to increase the yield and try to integrate some gluten genes to improve the gluten strength. Hopefully we can also improve the bread making quality. I think I will kind of briefly summarize. So in terms of deal with or address the challenges provided by, faced by farmers and consumers, processors, and the environment. So we need to understand the need, and then we can bring in the new variation either from the gene bank, like what we have done in Cassava, or we can create some new mutation, use EMS, use radiation mutation, or nowadays we have used CRISPR to create new variations, and then we integrate that into breeding program. So nowadays we have different tools. We have genome-based phenotyping. We have genome selection, and in Cassava we kind of redesigned the breeding scheme we started the hybrid cassava breeding, and in corn, we also try to kind of reshape the wheat breeding. Hopefully, we can create some new Semmendorf genes can overcome the setback of the green revolution genes. So I think that's all I plan to discuss today, and happy to answer any question. So it's pretty broad. If you have any detailed question or specific project, happy to discuss, happy to answer. Yeah, go ahead.
Do you have a sense of how common the RHT25 mutant alleles are in North American germplasm?
Yeah, that's a good question. I believe in the gene clone paper, they evaluated the allele frequency, and there are also several different alleles, right? So that's A, B, D, E, F, even different alleles. I don't have I don't have the frequency number in my mind. Yeah, I can quickly check the paper and find the number to you. And also probably different allele might also have different effect, but there's no deep study to understand to study their effect. So, and regard the RT25, I think the effect is pretty moderate. So we are thinking RT25, the plus might be not enough to short the plant height and to reduce the lodging. So that's why we are working on several genes, and hopefully we can find a combination. Yeah.
There's often a trade-off between nitrogen use efficiency and protein. So I'm wondering if you're also looking at nitrogen uptake efficiency at all.
Yeah. So that's for wheat production, that's always critical. Farmer get penalty I believe, if the protein is not high enough. Yeah. So come back to your question, the nitrogen use efficiency. I think for us, so here we have the model. We can predict the nitrogen percentage or nitrogen concentration and also biomass. So if we time these two factors together, so we consider is nitrogen uptake. So the time we evaluate the nitrogen percentage is during the flowering time. So we are thinking this, and I mean, so this probably it will be the peak time the plant accumulate the nitrogen in the plant, in the leaf, we can see. So we can compare the different lines in terms of amount of nitrogen uptake. So we can see we can use this to evaluate their nitrogen uptake efficiency. That's my current understanding. Thank you, Eugene.
Yeah, I believe you have this 3D is 5 or 15 later. Do you know what variety to release in that combination?
With 2, right? I think I don't have exact number, but I think it's not very high. So most is either or. So this is really, yeah, really stellar. Even in our program, we always said we need to have 2, but in the release variety, central red, central white, it's only have 1. So that's yeah, I try to fix this problem. Yeah.
Is there any kind of coordinating body for breeding between, say, IITAs, CIAT, and BRAPA, and Asian organizations?
Yeah, that's a good question. So in terms of cassava breeding, so in the CGIAR system, CIAT has a breeding program. IITA in Nigeria has a breeding program. And in terms of national breeding program, you mentioned Brazil, and BRAPA is the biggest one, and also Thailand is another very strong breeding program there. In terms of coordinating their breeding effort, during my time, it's mainly under the Next Gen Cassava project. So I believe I just heard some news several weeks ago. So there is a global cassava breeding program formed. It's mainly about CIAT and IITA in the CG system. So they coordinate their breeding effort and try to optimize their resources and try to make breeding more efficient. Does this answer your question? Oh, yeah. But in terms of currently with national program, since there is no funds to work together, it's mainly kind of conversation or capacity building. So this is kind of the jump sharing is mainly this type of collaboration with national programs. Then there might be some project like if the TDDI is a company or the organization in Thailand is very strong in support cassava, it might support some breeding effort also with CIAT. So there might be some that kind of project like a waxy starch, like CMD resistance, cassava mosaic disease resistance. There might be some kind of local collaboration. But globally with national program, there's no very kind of good structure on that. But in terms of CG system, I believe there is more structure now compared with several years ago. It's kind of separate. Rex.
Xiaofei, your career has taken you to different places around the world, and I was just curious, what attracted you to working in Colombia? To go, oh, I'm okay. That's good opportunity to move to Colombia to see it there. And then, what were some adjustments, challenges, opportunities, pleasant surprises that you found with your career switch at that time?
Yeah. So thank you very much, Rex. So I think working in Colombia, I believe if you have never been in Colombia, so you might have some bad picture in your mind. But so I worked in Colombia for 5 years. So I stay there. I think it's a very friendly country. And also very important, the center is international center. So we have broad collaboration. So with Latin America, with Africa, with Asia. So I think that's the major part. I really enjoy to work there. And also there is a very strong national program. I believe anyone familiar with the CG system. So the CG system have very supportive team. So even now, if I left CIAT, the new breeder coming in. So the team on the ground is the same. So that's very stabilize the system. So I think the position at CIAT, I think that's the best position I have ever been. So in terms of its mission is impact and again the collaboration. I think so. This is kind of the major thing really attracted me to join CIAT. To be honest, before I joined CIAT, I also know very little about CIAT, know little about cassava. But I work on sweet potato, so that's where I build that connection on that. So,
how long does it take you to become familiar with a new crop in your experience?
I think from intermediate wheatgrass to sweet potato, that's a little bit shock. But from sweet potato to cassava, it's a little bit easier. So it's both colonal propagated. So in terms of the adjustment, so it's work at CIAT, I pretty enjoy that. It's mainly because of the level of support there, and also I have 1 year overlap with my predecessor, so that's also give me pretty good. Hope I answered your question. Thank you, Rex.
So, quick question. You were talking about reducing gluten in the wheat breeding in terms of like, I'm sure customer base that could benefit from that could be people with like gluten intolerance, gluten allergies, and celiac. Do your breeding goals like, are they going to create a product that's potentially, I guess, usable by any of these different gluten levels of allergy and intolerance?
So yeah, your question is about the variety have different level of gluten.
Yeah, so, because the idea of reducing gluten is to
Yeah.
increase the customer base that can then use these wheat products.
Yeah.
I'm trying to understand if there's different strategies involved based on different levels of gluten intolerance or allergy.
So again, I think I have one sentence to see. It's very hard to measure the gluten allergenicity. So this is mainly kind of based on the scientists work on celiac disease. So they identify some epitopes which has large effect on gluten allergenicity. So now our research is mainly based on their result. So we try to eliminate those genes, and then we can, in theory, it reduces the gluten allergenicity. So now I believe, so this is part of Jorge Dubucasi's work. So he collaborated with some scientists, I believe, in Australia. So they are going to do some, I don't know whether it's human test or not. So we are going to share our sample, which with several deletions, and share. So those with flower sample, we are going to share with them. So then they are going to test whether there's any real gluten allergenicity reduction or not in our product. So yeah, we don't have any solid answer on that. So now it's still the first step. We develop those varieties, those germplasm. So in theory, it will reduce the exposure of gluten to the population. So this is kind of the big picture. So if, or the big theory, if the population have lower exposure on gluten, so the onset of celiac disease will be slower or will be less in the population. Yeah. So, but in terms of scientific evidence, we are still working.
Xiaofei, I have a question about it's a very exciting initiative, the hybrid cassava breeding program. And I'm curious about how, when you are starting from trying to figure out what the heterological groups of pattern looks like. How did you make those crosses? You select 30 founders? Did you just make a full allele, half the allele, or you did look at genetic, their genetic existence made the 30 crosses?
I don't think I have ever reached to that stage to develop the heterotic group yet.
Okay.
So I think so my time at Seattle, we just started the selfing, and we even studied the inbreeding depression. The inbreeding depression is mainly from the small population, the three progenitor we developed that. Yeah, so in terms of heterotic group, so this is just this is just some thinking or some theory. For sure, we are going to consider the diversity, and yeah, and then based on their family performance, then we can decide whether we can divide the 30 or even 20 of them into two pools, and then we can build up the heterosis. So I believe in reality there will be a lot of challenges, but yeah, so this is just some roadmap we propose. All right.
How do you manage the cassava gene bank?
Oh, that's a good question. So that's all tissue culture. So cassava, so the seeds is different from the plant, right? So it's more than 6,000 accession is managed by tissue culture. So every is 6 or 8 months, you need to transfer the tissue culture to a new medium. So that's a lot of work to manage that. Yes.
Questions? Not, thank you very much.